Ça marche?
Do you want to know that there are some... No?
Okay, transition. Okay.
Are you good on Matix? You have everything, eh?
Yes.
Okay.
I'm just listening to that. I will do the check of the YouTube. Let's go to Matix.
Okay, let me know if you need me. I will be with you.
Thanks a lot, Tim. Test sound. Test sound.
I don't know for sure.
Okay.
Tim is here for fixing the thing, so you can ask me. No,
Yes, I'm
I
trying.
didn't, I just realized that I didn't adapt the
Okay.
schedule, so
Yes.
when
Give me a second. I just try that.
Hi, Dr.
I'm sorry I cannot give you too much attention
No worries,
now.
no worries, no worries. I'll just settle in here.
Voilà.
Merci Jules.
Good morning, everyone. Good morning. So we will start this first session. During the session, as we already know, I will ask you to leave the room in between the presentations and to try to be quiet as much as we can.
Yes, can we check on Matix because I cannot find this? This is it.
Yeah, that's the
the title of your
presentation.
presentation.
Yeah, although there was a problem in Matix and this is the new title and it didn't get uploaded. I also had an abstract.
Okay, so you
But
this is the
the-
correct one. Okay.
Yeah.
So your name is
Everyone.
Yeah.
So for each presenter, it's 15 minutes of presentation and 30 minutes of question, and then 2 minutes to move to the next topics. Is it okay, Juan, if at like 2 minutes before you finish, I sign you like that? Okay, the floor is yours. We
Thank you.
listen to you.
So hi everyone, today I'm going to present to you my thesis project from the new revised title Fishing for Answers, where we're going to explore the trophic and spatial variations in contamination and nutritional value of coral reef fish in the Southwest Pacific, with a case study in New Caledonia. So my thesis was done in partnership with the IRD at the Mixed Research Institute of Entropy in New Caledonia for Project Tonic and under the supervision of Laurent Vigliola. So first, let's set up the context of malnutrition in the Pacific Islands. So let's try to understand the worldwide and local context. So malnutrition is a global issue affecting millions of people worldwide. It includes many disorders, from extreme hunger to obesity. Let's start with the quote. 44% of countries worldwide suffer from acute malnutrition issues. That really sets the worldwide context. And now let's set the local context. So here you have a map of the economic exclusive zones of the Pacific Islands, countries and territories, with a slight emphasis on New Caledonia, where I study. So due to an ongoing dietary shift from a historical low-fat food diet of the Pacific Islands to a more modern processed foods, we have a shift to high obesity rates and high prevalence of malnutrition in these Pacific Islands. To tackle that, in 2015, the United Nations set up goals, sustainable development goals, to provide a blueprint to solve these issues. And what I focused on were mainly 2, 3, 12, and 14, respectively aiming to have zero hunger, ensure good health and well-being for all, responsible consumption and production, as well as sustainable life underwater. Then, now that we introduced that, we can introduce the importance of fish in the Pacific Islands, and mostly the health benefit and risks of fish. So fish is a staple food of the region. It's really important for the culture as well as the food security and development of these countries. That's why they have really high consumption, up to 4 times the worldwide average. And fish constitute up to 90% of the animal protein intakes in this region. Of these fresh fish, 70% of the species consumed are coastal reef fish. And this is very good that they rely on fish so much because it's really a cornerstone of a healthy diet, mostly because it's a source of both micro and macronutrients, so essential nutrients, vitamins, as well as proteins, fatty acids, and carbs. And this is fish is something we cannot overlook to build sustainable and quality-oriented diets for the future. However, due to increasing and ever-growing anthropogenic pressures and pollution inflows, that may lead to human exposure to contaminants due to fish consumption. Therefore, fish represents a trade-off between health risks and benefits. So, before thinking about what we can do to remediate or at least study this issue, let's take a step back to have the background of the study and mostly the study sites. So, New Caledonia. So, New Caledonia is a prime diversity hotspot. It is home to the largest lagoon in the world, 31,000 square kilometers. It's also home to the second largest barrier reef just after Australia. It is also impacted by both natural and human threats because it is the largest nickel ore reserves in the world. It's home to 25% of the nickel laterites worldwide. It's also the third nickel extractor and producer in the world, so very high pollution, both from natural and human-driven reasons. So there you have a map of the main mines and main rock outcrops that occur naturally and also were dug as open cast mines. So to remediate to that, we had some research questions. What are the variation in nutritional quality and contamination of fish across space and species? And how do we quantify that trade-off that I said before about the health risks and benefits of fish consumption? What we expected to see were significant differences among species and trophic levels, with also major variation in contamination between sites. So now, let's present the methods. So first, the site selection. How did we select the sites? We applied the methods of ceremony, which basically constitutes a hierarchical clustering of 10 environmental descriptors, such as temperature, salinity, sea currents, as well and generating groups of similar environmental properties called ecoregions. These, so you can see there we computed that, and it got us 3 distinct ecoregions. So the southern lagoon, as you can see in red, the east coast and northern lagoon in blue, to the right side in the north, and the west coast in green. So we select from previous knowledge we have in the field and from previous studies, we selected 8 sites all around New Caledonia, with the capital being Nunula and all around. Although due to unforeseen weather events and political reasons, we could only focus on the 4 sites in the south, so these ones. For species selection, we based on the Institute underwater visual censuses for abundance and biomass to carefully select 14 coral reef species to have a representative of these assemblages of New Caledonia. So you can see the fish there. So as for laboratory analyses, we analyzed the concentration in heavy metals using inductively coupled plasma, either mass spectrometry or optical emission spectrometry. So oligoelements, which are usually in low concentration due to their name, analyzed by adjuvant ICP-MS, so 12 of them, which you can read here. And micro and macro elements, which tend to be higher in concentration, were analyzed by a Perkins-Elmer ICP-OES. But due to complication with the Institute's ICP-MS, the analyses were done externally by a laboratory team in Adelaide, Australia. Now, let's switch to the most important part, the main findings, what we found. First of all, we found a negative correlation between nutrients and contaminants. So as you can see with this correlogram, showing positive correlation in red and negative correlations in blue. The significance of this correlation were assessed with the Spearman correlation test. So this shows that we have a significant correlation between, negative correlation, sorry, between essential nutrients such as phosphorus, potassium, and selenium against all of the metals, and we as well have significant positive correlation between the metals themselves. So the metals are positively correlated due to their origin from natural rainfall runoffs on natural outcrops. As I mentioned before, the natural occurring peridotite. Nonetheless, the reasons of these negative correlations need further investigation. We didn't find reasons for negative correlation between the nutrients and the metals. This is a very original result, but previously overlooked in previous research due to the research failing to consider both contaminants and nutrients at the same time in the same design, because heavy metals and nutrients have different orders of magnitude in terms of concentrations. That means they require different tools to be assessed, so mainly ICP-MS or ICP-OES. So the second results that we found were trends between species. So these are from the Permanova test we performed, of which the results you can see here, we had a strong significant difference with the high fit of the model, with a mean R-square of 55%, which is good for us, for all but one element, vanadium. From the right graphs, you can see that the difference between species. That's all the species, and you can also see from the slide I mentioned before that when there's a high contamination, there is a low nutritional value and inverse. So, what are the possible explanations for these? We have variation in exposure from life history. That means fish live in different areas. Maybe they eat different prey, so we might induce different contamination. Also, the position in the food web, so the trophic position, can highlight potential bioaccumulation or magnification, or conversely, the opposite, bioreduction. So these results really highlight the importance of considering species when exploring other sources of variation. That's what we did in the next finding, a spatial variation. So we performed a Permanova test using the site and species interaction, of which the results you can find here. So we showed significant results for 6 elements, as you can see here. So phosphorus, selenium, arsenic, copper, lead, and cobalt, with a good fit of the model. Again, 51% is good for us. In the right, we can see 3 graphs, of which I'm not going to explain now. Spatial variation, when the first 2 contaminants are is proportional to the site's impact characteristics. So evidently, when there is a higher contamination, the fish flesh are going to have higher concentration in contaminants. But the opposite is true when nutrients are inversely proportional to the site's impact. When there's a higher impact, the nutrient levels are low. So as you can see, it's decreasing. However, the significant differences between these sites from the post hocs that we performed here are only marginally significant, up to the threshold of 10%. So now the last findings that we had was the potential risk for humans. As you can see, this big table, it's a bit full, but I'm going to try to have your attention on the green square. So this is the potential risk assessment based on the toxicological data of the high food authorities. So what we are looking at is this one, the ratio, and when it's below 1, that becomes a problem. So we can see that only arsenic is above that threshold, above that contamination threshold, dangerous for humans, but this is only total arsenic, and from this total arsenic, only inorganic arsenic is toxic. So AS3 or AS5. The organic form of arsenic, arsenobetene, is the most prevalent form in fish, mussels. Less than 1% is inorganic arsenic. And also the provisional tolerable intake of arsenic has been withdrawn since 2009 because there was no longer sufficient data on this. Okay, now let's switch to the conclusion. So our principal results. So we found a significant, strong difference between species, proving that it is really important to consider whole reef assemblages to visualize changes in contamination. The second one was we found marginally significant differences between sites, showing that fish have a higher contamination and lower nutritional value when the site is more impacted. The third one, we have a significant negative correlation between essential nutrients that opens an interesting perspective for future research, mainly the molecular approach on that. We have no risk for exposure to heavy metals for humans when consuming reef fishes in New Caledonia. The benefits for fish are far outweighing the risks of their consumption. So lastly, the critics and perspective. So the political and sociological context impair the sampling collection, inducing cascading problems. So we had no unpopulated sites being sampled. Mostly we only sampled these sites and not these ones. And there's more population in the west and the south of New Caledonia. And so if more sites could have been sampled, it could have given us a more global idea of the distribution and contaminants and nutrients at the scale of the whole archipelago. With more replicates per sites, we could have allowed us to have significant results on the spatial variation. And for the perspective, we highlighted two main ones. We could have predicted the values of nutrients and contaminants based on environmental parameters. So the ecoregion I calculated could have been used by boosted regression trees to predict these two values. And last perspective is to explore this original results that we have, the negative interaction between nutrients and metals at the ecotoxicological level. So these are my references in order of appearance. I know it's not in alphabetical order. And thank you for attention. And I wanted to thank everyone that helped me during this thesis.
15 minutes. Perfect. So we have 13 minutes for question. No? Do you want to ask a question?
Yes.
Don't start.
Probably because we have too many.
Too many, okay.
I can.
Okay.
Okay. First of all, thank you for the presentation. Nice work. Nice presentation. First question. Why the change of title?
The change of title was because the social context in New Caledonia is quite difficult, so we had to reduce the number of sites, and therefore we couldn't do modeling anymore. There was no large enough scale, so that we changed to more quantitative comparison between sites. So that's mainly why we changed it.
Adaptations to problems.
Well, that's it, exactly.
And that is coming my second question. Complication in the measurement, and you did the analysis in other place. Which kind of complications?
Basically the ICP-MS in New Caledonia just ceased to function. And we had problems, so when I landed in New Caledonia, they told me that the ICP-MS died. And there was no more in New Caledonia. There's only one. New Caledonia is such a small country, and there's only one, so we had to externalize.
In Orlando, when something like that happened, we always say ecothic. Sometimes it happens, and it's important also to adapt to that situation. It's okay. Which is your participation in the work?
My participation is basically everything but the laboratory analysis. So I went in the field, we collected the species, I went in the lab, started processing before sending the samples in Australia, and then did the analysis myself and the discussion and the redaction as well. So basically all but laboratory analysis.
Do you think that there's any metal missing?
Yes, I expected that question. Mercury.
Yeah.
I know it is missing.
Of course.
Although at the Flinders University, at the external laboratory, they said to us we need separate tools for mercury, and we did not have enough funding to do either to do both mercury and the other metals. So we chose to have take data. Maybe I know it's really consequent metal that we need to study, but we decided to study the most that we could.
And it's a good. I mean, it's really nice. It's not, but always the mercury is always.
That's true.
Something that we would like, and something that we know that is always problematic.
In particular in New Caledonia.
But problematic also for the measurement as well, because
when we also
I'm not chemist, but the chemist always, when you ask for more, always say yes, I need more sample, more tissue, and always is a lot of. So it's always difficult for us.
Because New Caledonia is such an original geological context with an abduction from the Norfolk Ridge. We have like peridoted outcrops, so it's mostly nickel, cobalt, chromium that is being washed out in the sea. So we wanted really to see the effect of mines on the fish and on the human consumption.
The mines were nickel.
Yeah, nickel mines.
Do you think that nickel is dangerous?
Yeah, but it's an essential metal. So at some point it's useful for the body, but there comes a point where it becomes toxic, and so there is that problem because it's such big quantity of nickel in New Caledonia that it might become a problem. Though on this slide it becomes not a problem, so we need to have 248 times the actual concentration for it to become a problem. So no, it's not a problem.
The last one, if I can. Yeah, okay. In the conclusions, I guess that you say something like it's important to take into account the whole reef. Do you agree, or could be any clue species?
We could always. I know in the bibliography they usually use the top predator of New Caledonia, which is a species of grouper, as a bioindicator. But there's such variability between species and also between individuals that I think it's really important to have a more global idea.
Okay. Thank you.
Yes. Thank you very much for your presentation. I'm convinced that it's a topic that is very relevant as seafood becomes more and more contaminated. So I would like to have your thought on why did you choose those three elements, P, K, selenium, as to highlight the benefit of fish, and what's your thought on that? And why do you call that, nutrient, and the link between nutrient and benefits for human consumption?
So we chose to also, this is taken from the literature. We have also, as you can see, I've like divided into three categories. This comes from the literature, and basically this is everything else than contaminants, that the ones that are really important for, I don't know, phosphorus is essential for basically life, so adenosine triphosphate is always phosphorus, and also it's in really large concentration and essential for the human body and always inside of biological functions.
So do you think it's enough to assess the benefit of fish?
Okay, I think for essential nutrients it's enough. Though to have the whole nutritional value, I don't think it's enough. As I presented in the introduction, we have so much micro, so we only focused on this part, so there's also vitamins, fatty acids, which are really important, proteins, carbs, and well, also that could have taken another thesis to study that, but I wanted also to do proteins, but it turns out we didn't have time to do so. I really thought it was really important, but we made some compromises.
Okay. Okay. Can you go back to the slide where you said that there was a positive correlation between a negative correlation
yeah,
between them? How can you explain first of all the fact that there was less what you call nutrient in the degraded area?
So, less nutrients in it.
Because if I understood well, there was a correlation between the environmental degradation.
So yeah. When it's high contamination, there's low nutritional value. As I mentioned here, we don't have a lot of idea of what could have caused that. As I mentioned, it's an original result. It hasn't really been investigated in the literature. So we are kind of moving carefully to not make crazy assumptions. And I'm sorry, but I can't really answer your question. And my thought was to go deeper into the ecotox level, at the molecular level, to see what is happening, if there is like antagonism between these metals against nutrients, but we are not.
Okay, still
well,
and what does it mean? Metals are positively correlated due to their origin. Positively correlated with what?
With themselves.
With themselves.
Well, so for instance, we can see that nickel and chromium are really correlated, so 0.85 correlation, so really strong correlation, and it's also significant on the correlation test that we did. So basically, the rainfall, the tropical rainfall on the natural rocks, the natural occurring peridotite, is washing out a lot of metals, and they are washing out at the same time, so they are positively correlated.
So meaning that when there is one metal,
well,
we will
there's
find
another
one.
well,
okay, it's contamination. Well. That's okay. And on the slide 7, last question. So do you consider when you did your analysis? This is when you have all the different fish, but when you do the correlation, it was taking all the fish together,
well,
correct?
Yes.
Okay, and this is where you believe that there might be a link that you would need to take into account the trophic level?
Yes.
Okay, thank you. Much more time we have.
So, congratulations, very nice presentation, very clear. Okay. We don't read the report in advance, so that means that you did quite well. The presentation, very easy to understand. Okay, some questions on about the selection of fish species. I guess that you go there. So, is the fish that you analyze is because you catch or you fish them, or you go to target in these species?
No, we really, I don't know if I'm answering your question right, but yeah, we really aim for those fish. So we aim for those 15 reef species to have like the true, more like a classical assemblage of the fish. However, in New Caledonia, maybe there's like some micro-endemism of fish down south or up north, but we try to aim as to the most general fishes and also maintain from the lower trophic levels to the higher trophic levels. So we try to have like that classical assemblage all around.
Related with what you had mentioned right now, I have another question, is that did you see any difference in metal accumulation according to the trophic position of the species? That because I think that the data that you show is integrative. As I know, I mean that it comes from all the species together, the values that you show, or you differentiate the species by species.
So this is all the species grouped together, and this is the only side where they are differentiated. I have some annexes afterwards. So this is the trophic index. So basically the trophic levels. So we can see that they are different, but they are kind of overlapping each other. It's a bit difficult, so that's why I didn't include that and didn't go to that specific way. It's because we have a lot of overlap and I couldn't really explain all of it. So, and it's so variable that it's really hard to really infer something that is true for all of the trophic group. So we were really careful on that, and we tried mostly to go species by species more than the trophic level itself.
Okay, and for example, this classification, okay, so do you think that is because it's a limitation with the number of individuals you got for per species, and so maybe if you had the opportunity to...
Yeah, more replicates would have been great.
Yeah, because...
it's always a question of money and time.
Yeah, sure.
So, yeah, we had... so that's one of my critiques, actually. If we had more replicates per site, it could have allowed us for significance and for more, maybe more clear patterns to maybe infer.
It would be nice just to see if there is some kind of trends, okay, between the basic species and the more the species that are at the top of the traffic. If it is some kind of fire population.
If that can help you, these are classified based on trophic index. So this is the lower trophic index, this is the higher trophic index. So I tried to do a bit, but as you can see, all of these are lower trophic levels, and then it's a lower trophic level, but see these four are the same trophic group, but this one is significantly higher than the rest, so it's difficult to have this, but also a really big variability always. So more replicates again would have helped us.
More
More samples, more.
Well, we will thank you for this discussion. We have now to move to the next speaker, but if some people want to ask you more questions, we will find more time. Thank you.
Just a small note, don't turn on the speaker notes because then the presentation is not online.
Oh,
it's a small note. Who's next? How many?
Yes, it does that. Yeah, it did that to me before. I did that. No, no, this is what it should not do.
No, it's okay, it's okay.
I did that last time. No. Where is
But now
Yes.
Wait, I'm just gonna check now if I see the screen. Yeah, should be fine. Okay, should be fine.
Okay, so we have our next speaker ready. And so we will start listening to Anna for 15 minutes.
So good morning everyone, my name is Hannah and I did my thesis at the University of Algarve on the exploration of Limbada crinoid for the phytoremediation of acetaminophen through in vitro plant tissue culture. I would like to start off this presentation with a quote of Sylvia Earle, stating no water, no life, no blue, no green, emphasizing the importance of water and emphasizing the importance of research done to combat the problem of water solution. One of the contaminants that has gained more attention for 2 years is the contaminants of emerging concern. These are micropollutants that have been detected in sewage recently and can potentially harm human life and the environment. Up until recently, there was no legislation on monitoring and reporting of these contaminants, but in 2022, the EU recognized the problem and changed its legislation demanding 80% removal. This led to more sewage being collected and purified. However, current wastewater treatment plants are not sufficient enough to meet these expectations. In my research, I focused on acetaminophen, otherwise known as paracetamol, because I did it in Portugal, and a study done on the Guadiana estuary showed that acetaminophen is one of the most prominent contaminants in the estuary. Once it enters the water body, it can bioaccumulate and potentially also enter the food chain, not only having an impact on the aquatic ecosystem, but also on us. And it also remains bioactive. Since traditional remediation methods are not specific enough or are too expensive, there is a need for new techniques that can improve the removal of not only acetaminophen but like contaminants in general. And one of those techniques that gain more attention is phytoremediation. Phytoremediation is a green alternative because it's based on solar energy and natural processes occurring in plant, making it more eco-friendly and cost-effective. It can be implemented in real-life scenarios, for example, through constructed wetlands. What happens is that the sewage, the effluents go through the wetland, and the plant should take up the contaminants, and then it can go to the estuary or to rivers and so on. In Portugal, Limbada crinoid has been used to end those constructed wetlands. It's a salt marsh plant, and it's native to the Algarve region. Because it's a salt marsh plant, it's adapted to being able to thrive in a harsh environment by, for example, being tolerant to high salinity and having a deep root system. And that deep root system can make it more easy for the plant to take up the contaminants. And that's why in my research I used this plant. So for the objectives, we first tried to set up a phytoremediation system. Once that was successful, we tried to evaluate the capacity of the plant to take up the acetaminophen, and then also evaluated the impact that the contaminant has on the secondary and primary metabolism. So how did we do that? The first step was to establish in vitro propagation using an explants of the Limbada crinoid that has been previously cultivated in a greenhouse. We we put these explants in sterile glass tubes containing a mist medium, and these were then maintained at a constant temperature with 68-hour dark cycle for 4 weeks. After those 4 weeks, we saw that enough roots and shoots were developed, and then we divided all the plants into 4 groups. One group containing the control, and then 3 other groups that were spiked with 3 different concentrations of acetaminophen, as you can see on the slide. After then 7, 14, and 21 days, samples were collected. We separated the roots, aerial parts, and the medium, and those were then kept in the freezer until we did the second step. For the second step, we developed an approach to be able to quantify acetaminophen in the growth medium. Herefore, we focused on the feature of acetaminophen being hydrophilic. We first had to pre-concentrate the contaminant in the growth medium by solid phase extraction. Herefore, we used a suprapoly HLB SE column with methanol as a reagent for the elution step. And then we did a high-performance liquid chromatography diode array detector to actually quantify the acetaminophen. We, throughout the HPLC, we changed the mobile phase to being able to separate the peaks that were found in the first trial better, and then we detected it at 243 nanometers. And then the final step was to actually measure the plant's resilience. Therefore, we analyzed photosynthetic pigments, osmoprotectants, secondary key metabolites, and oxidative stress markers. Depending on what compound we analyzed, we did different extraction methods and different biological assays. And then all that data we gathered was then analyzed using ANOVA and a post-mortem test. So now for the results and discussion, I first wanted to emphasize why we said that the in vitro validation was successful. First of all, like no plants died, which is good. We also didn't find any contamination, and as you can see on the slide, you see that a root has been developed of 4 centimeters, so we were happy and we used it. Then for the quantification, this graph shows the acetaminophen left in the growth medium for the different treatments and throughout different incubation periods. What you can see is that after 7 days in all the different treatments, there is a decline, suggesting that the plant can take up the acetaminophen. However, we didn't use a negative control with only acetaminophen, and we also didn't analyze the biomass, so we're not 100% sure, and maybe the decline is because of degradation of the compound. Then, for the plant responses, the first compound we analyzed is MDA. MDA is produced because when plants are stressed, they produce something called ROS, and that will cause lipid damage, and one of the metabolites that increases is MDA. Before I go into the results, like every graph you will see, we'll have on the y-axis the concentration of the compound measured, on the x-axis the different treatments. The different colors of the graph show the different incubation times, and then the letters above show that the capital letters show the differences between incubation periods, and the lowercase letters show differences between treatments. So what we expect is that with an increased incubation time and with higher acetaminophen, we would expect that the MDA levels increase. However, this is only visible in aerial parts for the plants treated with 1 milligram per liter. So maybe the plants are already adapted to it, or the standard is too high, so we have to repeat it multiple times to be able to really conclude that. And then in the roots, compared to the aerial parts, the roots are like more stable, and they have also a kind of a lower MDA content. And my hypothesis was that the acetaminophen maybe could be transported to the aerial parts. However, we need to do more research to know that, because my results cannot confirm this. Then we also analyzed the photosynthetic pigments. We analyzed the carotenoids and the total chlorophyll. In the aerial parts, you see that at higher concentrations of acetaminophen, we see that there is a decrease on day 7, suggesting that the detoxification mechanisms a plant has to protect itself from stress are not sufficient enough. However, there is actually a boost on day 14. But that boost is temporary since we see again a decline on day 21. So or the plant is adapted to the stress of day 21, or the plant is too stressed, causing damage to the photosynthetic apparatus. And the roots then you see the same trend, but it's less visible, which is logical because the primary function of the roots is not the production of the pigments. Then we also analyze its osmolar protectants. The proline shows a similar trend as the sugars. And what you can see is that when the protein content is high, the proline and sugar content is low. And when the protein content is low, the sugar and the proline content is high. And that is because proline and sugars are used to stabilize proteins and are used to produce proteins. So that is confirmed by this. And then for the roots, all those thermoprotectants measured show the decline, meaning that in the roots, the proline and the sugars are depleted, but are not used for protein production. So maybe the plant focuses on other compounds to protect itself. And then finally, when plants are stressed, they also upregulate a pathway called the shikimate pathway, which results first into an increase in shikimic acid. But then that shikimic acid is used to produce phenolics like flavonoids through with an enzyme called phenylalanine. And that is what you can see in the aerial parts. You see that there is a decline in shikimic acid because the PAL uses the shikimic acid to produce phenolic compounds. And then in the roots, you see more stable levels of PAL with more fluctuating levels of CA. So probably that also will lead to more stable levels of phenolics. So when you then look at the phenolics, you see actually what I said, an increase in concentration. But you don't see an increase in flavonoids, and we didn't find any tannins, meaning that the plant focuses on other phenolics like probably lignin to protect itself. And then in the roots, as what I expected was that compared to the aerial part, there's more like stable levels. So what I can conclude is that the phytoremediation system that I established was successful, that the plant shows high remediation potential and also high capacity of being stress resilient. But there is a difference between the aerial parts and the roots. In the roots you see more stable levels, maybe because they are less attractive or because of the translocation. And the roots show more stress resilience by upregulating the shikimate pathway and the sugar and protein levels. But to understand why there is such a difference between the aerial parts and the roots, we have to look deeper into where the acetaminophen is distributed in the plant. So finally, I would like to thank all these people for having me. Thank you.
Thank you, Anna, for this nice presentation. 13 minutes, yes. So we have 13 more minutes for questions. Who wants to start?
Okay, yes. Thank you, nice presentation. I was thinking about the phytoremediation. Do you think that this phytoremediation a possible solution? I mean, is real solution?
I think it's a nice concept. Like the idea behind it is like beautiful because like you use natural stuff, but it's still really early and we have to do way more research to see if it's actually feasible because we have to have the plants that need to grow. Like there's so much other stuff that you need to be able to do before.
For instance, which should be the plan. I mean, I plant a lot of Limbarda that also could produce ecological problem as well if you are making a big station of, because you need a big station just to be able to phytoremediate somehow. So, what do you think?
Yeah, it's a difficult question.
That's why I'm here.
But yeah, can I think about it?
Probably is not an answer. It's not a good answer in any case because I know that it's so complicated.
Yeah.
But that's why maybe you could adapt as well the title because I should say that your nice presentation of stress produced by the contaminant.
But the thing is, it's like this is part of a really big initiative like called We Treat, and they really focus on trying to implement the constructive wetlands to remove it.
Okay.
And that's why I put phytoremediation.
Good. And then the next question will be you plant everything and then what you can do with the plant.
What you can do?
I mean, once you have the plant grow and it's eliminated some of the contaminant, then you have to manage as well.
Yeah, it's a lot. But yeah.
This is because we have some
idea.
we can have some project with phytoremediation as well, phyto and vermi remediation with polychaetes and that kind of things. And I don't know if we are generating more problems sometimes. I mean, it's like.
But it's a nice idea.
No, but it's really nice, yeah, of course. But again, with the title, maybe the stress produced by the contaminants, but the thing is that in the conclusion you put that it could be a good plan for phytoremediation. You know something about more contaminants? Could be also phytoremediate, or is just this specific?
We only focus on acetaminophen, but in the beginning, like I started in the first semester of this year, and in the beginning we said if we have more time, we can do more pharmaceuticals, but at the end, like we didn't have time. It would have been better if we did more pharmaceuticals. But
What is a micropollutant?
I forgot.
No?
No.
No, I don't have clearly the idea of micropollutant. I don't know exactly what it means.
Yeah, I was.
Or
thinking
I don't know
that
if
when
I
said
agree
it.
with the definitions. Okay, I think it's good for me. Thank you.
I have a more specific question. Can you go back to the first slide where you showed the first results? And thank you, yes, for your presentation, which was very clear. So this one, next one, sorry. So here you showed that you have accumulation. It was unclear to me.
It's not accumulation.
What's
It's like the concentration that the acetaminophen, like the concentration that is left in the growth medium. Like on day 0, we have 0.1, 1, and 2, and then after day 7,
It shows then that it was transferred to the plants. It's your indicator that is no longer in the water, so you assume it went in the plant.
that's an estimate.
Yeah. No, it's well, why not? And then you have then your result.
Here?
So you have never tested the acetaminophen in the plant? Then
No.
That's what I. Okay.
Yeah, because we didn't have enough biomass.
Okay, because I mean, one of my questions was why do you say it's successful?
Because, like, based on this, we say it's successful, and also from other research, they say that paracetamol is stable. So we expect, like, we think that the plant takes it up, but we cannot be 100% sure.
Then you mentioned mentioned that the acetaminophen is a stress to the plant. Why do you say it's a stress? It's based on the literature?
Yeah.
Based on the literature.
Yeah. And also, like here you see that, like normally when plants are stressed, like the MBA level increases, and that's what you can see here. Like throughout the prolonged exposure, you see an increase. But for the others, it's not really significant. But I think it's because, like you can see here, there is a really big standard there.
And the last question, if acetaminophen bioaccumulate, why do we find it in the environment? What is the source of contamination?
Source?
The source, why do we find acetaminophen in the...
It can be because like it flushes through the toilet, it can be because
how can you get,
through most of the time through human activity.
Okay, but so it does not bioaccumulate in human body? If it gets peed or poop, it gets eliminated from the human body. Does it mean that, because if we consider that, I mean, you have measured your acetaminophen in your environment, and you have not seen it getting back, so it meant that it bioaccumulated in the plant. What do you know from the literature about bioaccumulation of acetaminophen? Mammals versus plants, would there be any specific value?
I didn't see anything on that.
Okay, thank you.
So, thank you for the presentation. So, it's very clear, and so you explained quite well all the concepts. And I mean that the results are also quite clear, explained and described. But I have some doubts about the effects that you present, considering your methodology or your experimental approach.
Okay.
The thing is that what I understood is a static experiment. I mean that you put the water contaminated and you maintain that for 14 days. You don't replace the media.
No.
Okay. And but go to the same slide where you show the levels in water of the pharmaceutical. Okay, that is one. So that means that after 7 days there is almost nothing.
Yeah.
Okay, so in the first week everything is gone, let's say, or degraded or whatever. So it's hydrophilic, so we suppose that, as we will comment, it's not bioaccumulated at high levels, so the effect should be something different. My concern is that some of the responses that you observe maybe after 14 or 21 days in some of your markers is maybe they are related because you have less nutrients in your media and not pharmaceutical effect.
Yeah.
I mean that some of the stress, because most of them are related with antioxidant capacity or oxidative stress, I mean in a stress scenario. So
yeah,
that's my concern of if really it's a contaminant effect or a stress situation of the experimental design.
Yeah, it's true.
Okay.
It's true. That was one of the remarks I talked about with my supervisor, that maybe we have to think about it if he wants to repeat it.
Have you measured the growth?
No.
No. Okay.
No, we wanted to do that, but then I made a fault. So then we did not do that.
Okay, no, it's something just to compare, to see on that, and it would be nice just in the future if you continue maybe to include other kind of, maybe earlier, I don't know, 3 days, 4 days or something like that. So in order to see if maybe there are some kind of responses earlier, because this degradation is just, I don't know, I can assume that probably the media was sterilized, so no bacteria there, because they tend to degrade. But that's all from my side.
Okay.
Thank you.
So we just have time for a short question, an additional question. So I would just ask you a quick question, so and then you're free to go. In fact, I want to pursue the discussion of my colleague. Given the fact that you have in the media, you don't have the contaminant anymore. When you look at the oxidative markers, how do you consider that the plant answers to the media? Do you think that when you look at an oxidative marker, it integrates the exposition within the plant? Like in the media, there is no stress anymore. So you discuss the evolution of the oxidation markers within the plant. Do you think that it's an integrated response, or do you mean that the plant responds? What you see is the immediate answer to the media. Like if you look at day 21, you should have no stress. There is no acetaminophen in the media. So what do you think you're looking at? The exposure at day 21. When you look at the markers of oxidation, for instance.
Well, at day 21, like we expect that the plant like got all the acetaminophen, and then just like because, and then yeah, I don't know. I don't understand the question like you.
Okay, so look at the experiments. After 7 days, there is no more contaminant in the media, but there is the plant. What is your assumption? The contaminant is still in the plant, and the response that you see is because the contaminant is within the leaves and the roots, or is it what you see is that the plant has degraded the compound anyway?
But that's like you can analyze that if you do a GCMS. You can analyze if there's like if the plant degrades and then you can metabolize. Because actually, what we saw is that the acetaminophen in the day 7 was lower than at day 21. But the differences were not significant. So maybe the plant exactly like degrades it and then the metabolites go back into the medium, but like, we're not sure about that.
And there is something that I was surprised. You rarely compare the responses to the control.
Yeah.
Why?
Because it was super difficult, and I like in my manuscript I did do that, but then here they told me to focus on like specific stuff and make it easier to explain it.
Okay.
Thanks a lot. Talking shit.
Okay.
Sorry.
Is it okay?
Yeah.
Okay. So we start our last presentation.
Thank you. Good morning, everyone. I'm Ivan Patrick Buchanan-Tuwala, and on behalf of my research team at the University of Bergen and our collaborators, I am here to present the result of my master thesis project titled From Deep Sea to Humans, Discovery of Novel Promiscuous Enzymes from an Arctic Deep Sea Hydrothermal Vent, and Novel Function of Human Butyrylcholinesterase and Depolymerization of Plastic Substrates. This study is supervised by Dr. Renar Stokke and co-supervised by Professor Ida Helen Steen and Dr. Katarina Sass. My presentation will start with an introduction, followed by materials and methods, results and discussions, conclusions, and acknowledgement. The slides are numbered and the references are written as footnotes. To begin with, discarded plastics can be found in almost all habitat, making it a ubiquitous and a persistent pollutant, and it is projected to further increase in the decades to come. In fact, in the marine environment, around 27 metric million tons of plastics are being discarded that can be found from coastline to pelagic waters and even reaching down to the deep sea abyss. And most of the plastics that can be found in the marine environment are actually polyethylene terephthalate, or PET, because this polymer is used to make plastic bottles, single-use plastic bags, and even in the textile industry. So we really need to find solutions on how we could address this, since it's jeopardizing marine biodiversity and even putting a risk in the public health in the bigger picture. Currently, plastic recycling methods face predicaments, since we do mechanical and chemical treatments or even incineration so that we could repurpose or recycle plastics. But then it has been shown that these approaches contributes to a very high emission of greenhouse gases, high carbon footprint, and even related to or correlated to climate change. Alternatively, bio-based solutions such as enzymatic degradation of plastics are now currently in the spotlight, thereby giving the rigorous enzyme discovery so that we will be able to find enzymes that could potentially close the plastic recycling loop, making it or driving it more into a sustainable and a greener approach. Of course, nature always finds its way. In 2016, Japanese scientists discovered a bacterial strain that could degrade and assimilate plastic monomers and produce energy out from it. Here is a micrograph of Legionella sakaiensis on a PET film. And if we look on the molecular pathway of PET degradation, this bacteria has molecular machineries that could actually degrade the PET by the group of enzymes called phytases or cucinases into two monomers, BHEP and MHEP. And another group of enzymes called amylases can be also used to degrade MHEP into a monomers of TPA and ethylene glycol. Now, these two ending monomers is very important because it can be used for the biotransportation or biotransformation rather of bacteria for biotechnological approaches, or even use these monomers to make a new PET polymer. But if we look on the landscape of PET degradation pathway, there's still a missing link. And that is a group of enzymes called BHEPases that will degrade the BHEP compound into MHEP and then further down into TPA and ethylene glycol. Most of the plastic degrading enzymes currently being reported are from prokaryotes or bacteria, some are from fungi, and some from algae. But most importantly, no reports up to this date have been conducted to examine if higher vertebrates could have plastic degrading enzymes. Therefore, this study aims to employ a bioinformatics-assisted metagenome in silico enzyme mining approach to discover novel plastic degrading enzymes using in situ enrichments of the Ager hydrothermal vent at the Arctic mid-ocean ridge. Specifically, we wanted to try a new approach in the enzyme discovery that utilized structure-based homology for the selection of candidate enzymes. Our candidate enzymes, we wanted to produce it in the laboratory, and also through a heterologous expression using and a protein purification using E. coli cell system. We wanted to check the functional screening of these enzymes for the active degradation on plastic agar plates. And also we want to validate if our candidate enzymes could actually degrade PET in a solution. And we want to elucidate the sequence feature and structural features of our candidate enzymes, comparing it to reference enzymes. To give a context in my study, the enrichment at Ager vent field was conducted at the Amur. So here is the series of hydrothermal vents. My research team did the sampling at Ager, wherein they had this specialized titanium rods with sediments amended with different substrates, and it was incubated there for a few days, retrieved, and those sediments were used as a starting material for the enrichment in the laboratory. And then the enrichment here was subjected to metagenomic sequencing. All of the open reading frames were annotated. And then my thesis specifically started with the annotated open reading frames to do the enzyme mining. But if you look closer for the structure-based homology enzymes, we use a high-performance blast P analysis of all the open reading frames using a established plastic degrading enzyme database called PASI, meaning that we want to check if these putative ORFs have a hits on this database, and then we look on the pentapeptide motif inspection of all the ORFs. We specifically look at the GXSXG pentapeptide motif, where X could be any amino acid residues. And then we modeled all the structure of the candidate enzymes and we used all of the available crystal structures in PDB databases, and we modeled as well the predicted structures from the PASI database, and we utilized the crystal structures or protein structures available by AlphaFold. And by that, we did the structure-based homology by network analysis and visualize the different networks where we could find potential candidate enzymes. Once we selected those candidate enzymes from the metagenome assembled genomes, we then proceed with the enzyme production and functional screening, wherein we did gene synthesis to optimize it in an E. coli. We expressed it in the laboratory and did a two-step purification, IMAC and gel filtration. Once we get the purified enzyme, we dialyze it to change the buffer into a working or suitable buffer, and then check first the functional screening here on an agar plate with plastic substrate with BHET, PCL, and PU, because these are the available plastics in the laboratory. Now, I want to repeat again that BHET is the monomer from PET. PCL is one of the new biodegradable plastic, or we call it polycaprolactone, and PU is polyurethane. Once we confirm some activity here, we then wanted to do the in vitro validation using PET assay and BHET assay at different time points. For PET assay, we use a low crystalline amorphous PET powder, and for BHET, we use a BHET solution to do the validation. It was quantified using UHPLC, and then we look at the sequence features, and we did a structural superimpositions of our candidate enzymes, and specifically looking at the catalytic sites, and do the data analysis. Now for the results, putative plastic degrading enzymes are present at agar hydrothermal vents, as shown in table 1. Here we are able to screen 6 enrichments for a total of 500,000 open reading frames, and all of these were then categorized the classification using the PASI database, right? As you can see here, enzymes that are potential to be purases were the highest. The rest are evenly distributed, and no accounts for enerases, BBcases, for example, because up to this day, we still haven't found enzyme that could degrade actively on those type of plastics. Also, interestingly, we found that there are potential bifunctional enzymes in the screen ORFs as shown in figure 2. As you can see here, LDPE and PU foam are the substrate that mined with potential plastic degrading enzymes that could be promiscuous, meaning that it could degrade 1 or 2 more enzymes. And PET has the lowest number of bifunctional enzymes. And here is the 2 candidates, MAG7 target and MAG1 target, identified as candidate enzymes. Here is a snippet of the multiple sequence alignment. I just want to show here the pentapeptide I was talking earlier, showing a conserved motif with the known regional saccharinaceae and Vibrio gazogenis. And here is just some details on what is MAG7 target and MAG1 target. Specifically, MAG7 target has a 36.2 hit on the PASI database and is taxonomically similar to Pelagiobacterium sp. MAG1 has a 43.6 hit on the PASI database and has a taxonomic similarity to Parabocholeuria, meaning that the 2 candidate enzymes were not biochemically described yet. Now, here is the structure analysis or the network analysis using the new approach, structure-based homology, where we were able to identify a human enzyme, butyrylcholinesterase, co-localized in one of our candidate enzymes. So first, figure 3A is the cluster network. It's a structure network of MAG7 target. Basically, there's so many crystal structures that has a structure similarity with our candidate enzyme, but the most interesting part here is figure 3B, because this is where the MAG1 target with the human butyrylcholinesterase co-localized together, meaning they are in a one cluster, implicating that they share a structural homology. And here are the lists of the enzymes we use in the study. Of course, the MAG7 target and MAG1 target, and then we use PET6 from Vibrio gazogenes as a positive control, and also to have a marine-derived representative of plastic degrading enzymes. And we have used a recombinant and wild-type version of human butyrylcholinesterase provided by one of our collaborators. So the wild-type here was directly extracted from human blood plasma. So for the results of discussion, first we were able to produce our candidate enzymes as shown here in figure 4. Here are the SDS phage gels showing the from the start to the purification, and the candidate enzymes are labeled with a black asterisk, meaning that in E. coli system, the expression and the solubility of our candidate enzymes were successful, even the PET6 as a positive control. And here is the functional screen of the candidate enzymes, wherein it shows you the plastic plate agar, wherein the top portions are the plastics we use together with our control buffer, and the candidate enzymes from MAG7 target to the human butyrylcholinesterase wild-type. So here you can see that any changes or the formation of halo zone indicate a positive enzymatic degradation, showing that MAG7 and MAG1 target are both promiscuous for PCL and PU and inactivity on BHET. While PET6, of course, it has been reported to degrade PET, so it should be acting on a BHET, as shown here in a cloud precipitate, but it has never been reported to be promiscuous, and this study showed that PET6 could also degrade PCL and PU, as can be shown with the clear halo zone. And if we look with the two human versions of the human butyrylcholinesterase, a clear halo zone is observed on BHET and inactivity to PCL and PU. Now here is the PET degradation product release of our candidate enzyme over different time periods. The y-axis here shows the total PET degradation products in micromolar. And the enzymes, as you can see here, PET6 is the only one that is active, meaning that through time, the PET here is degraded into the monomers, and the candidate enzymes would not almost show activity. But here on the BHET assay, we can really see that the BHET is we use the human enzymes here to observe if they're able to degrade BHET following the plate assay. And as you can see here, that the BHET through time is reduced into MHET, and after 8 hours, it's reduced to TPA, meaning that the human enzyme can actively degrade BHET in a solution. And on the structural features, we wanted to ask why do they have these characteristics. Here is just a sequence feature of our candidate enzymes with a PET6 hydrolase showing the unique features and not conserved catalytic sites. And for the PET6, the human enzymes, it shows that it has a very conserved similarity to a known bacterial BHAs, particularly on the catalytic sites shown by red triangles. And then here is the structure of our candidate enzymes, showing the alpha-beta folding of a typical hydrolase, and the crystal structures of PET6, the pink one, and human butyrylcholinesterase in the green one. And looking at the structural superimposition, RMS value lower than 2 indicates a very high similarity, as we can show here that MAG7 target has a high structural similarity with PET6, but differences in catalytic sites. The same with our MAG1 target. As you can see, it has a low RMSD value and semi-conserved catalytic sites. But here in the human enzyme, comparing to a bacterial BHAs, very high structure similarity, particularly on the catalytic sites. So it could provide evidence that it has an active degradation on behead. And to wrap up everything in this study, we were able to identify 2 novel marine hydrolases from a deep hydrothermal vent, and we showed the promiscuity, particularly of the MAG1 target, MAG7 target, and PET6, and the lack of biochemical characterization might have influenced why they're not very active. And our new approach in structure-based homology were effective in identifying a human enzyme that had active degradation on behead, on both plate assay and behead assay, and with a high sequence similarity and structural similarity to a known bacterial behead Thesis. And we somehow wanted to show that this human enzyme has the potential to be behead Thesis. And before I leave the stage, we want to acknowledge all the individuals who help us and the funding agencies. And before I leave, I want to thank everyone for joining with me, or our team, in our quest to help make to help plastic recycling into a greener way, one enzyme at a time. Thank you.
Thank you very much for this fast and intense presentation, 16 minutes. So there is probably many questions to be asked.
Please do.
I don't know if
I'll start.
Okay.
I would like to know, when do we consider that the plastic is degraded, because you reach your aim? Well, first, thank you for, yes, again, very dynamic presentation, so that was very pleasant to follow. So yes, why are you trying to obtain TPA and EG, and what do we know about those compounds? Why do we consider that degraded? When do we consider plastic degraded?
Exactly. So if we talk about PET, because of course there are many different plastic polymers, but in this study we focus on PET. For example, there is a debate right now with the marine ecologists of, for example, a PET that is already in the marine environment, where plastic here, colony of bacteria are thought to be degrading the PET plastic. But on the chemistry level, plastic degradation, or PET degradation, is only considered if the polymer, the ester bonds, were actually split by the enzyme itself, and not by the degradation of the additives, for example, or other chemicals that can be found in the PET polymer. So here, it has been established that an active enzymatic degradation of PET is true and reliable if we can detect the monomers BN and MN, and even detecting the TPA and ethylene glycol, because those are the building blocks of our PET, which could be used later on into a different approach. So in this study, we use that kind of principle to say that an enzyme is actively degrading PET if those were quantified and validated using HPLC, because that is the standard method now to use and to confirm that the monomers of PET from a poly chain were broken down into these monomers.
Do we know anything about the toxicology of TPA?
That is a very interesting question, because currently there's no literature as of the moment about what happens next if we have TPA or ethylene glycol. What we know for sure is we could use it for upscaling or biotransportation of bacteria, because ethylene glycol and TPA can be incorporated into a metabolic pathway to produce energy. But in higher vertebrates, as of the moment, we still don't know what could be the effect if there's higher concentration of this in our system, knowing, for example, that the human butyrylcholinesterase was extracted in the plant plasma. So this research is a benchmarking of what we currently understood of how these enzymes is playing a role in higher vertebrates, or even in different organisms. But at least we now have a baseline that it has the potential. But of course, many studies should be conducted to further confirm its, yeah.
Out of curiosity, you have shown a lot of illustration of tertiary structures.
Yeah.
Where have you, and it looked like it's published, have you been publishing your work already, or is it just reference that you have used here? So meaning that those structures were already known and published?
Yeah. So for the two result crystal structure, yes. This one is the PET6, it was already published and available in the PDB. And this is also published by our collaborators. So meaning that these structures are almost as it is. But our candidate enzymes are still predicted models using AlphaFold. We could not do crystallization because of time constraint. Yeah, but since AlphaFold has a very high accuracy in predicting structure of enzymes, we could then use it later on to check the accuracy of the predicted model and the crystallized version of the enzymes. We have really a resolved structure of our candidate enzymes. But you're correct, Professor, the two are already published and we use it as a reference.
But you are showing here, this is the predictive model of, so that would be the new part from your presentation based on your research. Okay,
Yeah. Here I just want to give context of what they do. Yeah.
Okay, thank you.
I don't have. I don't know if I don't have too many questions, or I have thousands of questions. I mean, it's one tricky.
Yeah.
When I saw the title, I already knew that the presentation would be also intense. And the critic is perhaps too much information. But really nice organized. So, good job.
Thank you.
Questions. Wow, it's incredible. I have a lot of questions, but related with the toxicity, I'm quite skeptic about the toxicity of plastics. But well, no, we are not going to go in detail. Which could be the application? I mean, because at the very beginning you said that the degradation of the plastics, there are different methods. Could be this used as how?
For the enzymes. Okay, so plastic degrading enzymes, particularly in PET, find it so hard to react with like this, a PET product, because this is in a high crystallinity form. So we're not saying that enzymatic degradation is really the holy grail of how we recycle plastic, but it could be used or incorporated within the steps. For example, a PET bottle will be shredded into a smaller form, and then by that part, the practice now is it is transformed into a thread for textile industry, and at that step, we cannot recycle PET anymore. But if we integrate a part of the enzymatic solution in that cycle, we will still be able to retrieve these monomers that could be used later on for different approaches. So it's not a one-step solution. It still needs to be a holistic approach. Yeah.
I think it's okay for me.
Do you have some question or?
Yeah. So thank you for the presentation. Very, as they say, so very intensive and so a lot of different information, but very well explained. That's very interesting. So first question is, okay, it's a lot of work behind that. So all that was done by you?
Yes.
Or there was some, because I mean that my question
His lab like is here, you know?
you start talking about metagenomics,
Yes.
and then you go on identification of the target sequences, then you go in order to identify which kind of the protein, so then you do the assays, the in vitro assays, and so on. That for 6 month period is very, very, very interesting.
Thank you. Well, to give a context, the sampling was done before I even arrived to my laboratory. Of course, my research team is watching now in the Arctic because I'm doing the first time. I was fortunate that my proposal was approved immediately, and I was allowed to start my master thesis during my third semester at the University of Bergen. So technically, the bioinformatics pipeline year was conducted during my third semester, and the actual wet lab work started last January for a total of 6 months. But producing the enzymes to be soluble took me 4 months, but I did not give up because we need solutions. But yes, it was mainly done by me with the guidance of my supervisors and our collaborators at the University of Hamburg, because that's where I did the HPLC analysis. So you're watching, hi. Yes.
And for example, so because I can imagine when you started just taking the genomic data from this kind of course, so I can imagine that there are dozens of species contributing to that let's say, degradation and so and production of the DNA product. So are you able, just so you, did you able, or you are able to differentiate the species that have
Okay. Well, I cannot really disclose the species because it's currently being prepared for a different publication. But if we go back here on the similarity, okay, here. So MAG7 target is taxonomically similar to Pelagibacterium sp. Now, this group of organisms, there's no reports that they are actively degrading, but the family where it belongs, family, the name, Hyphomicrobiaceae, they were already reported to degrade plastic enzymes. Some of them from that family are even found in the deep sea. For MAG1 target, Parabulfolderia belongs to Parabulfolderiaceae family, and many of its species, or some of these species, were even reported to degrade PU, and what was shown in our plague assay. Some of them were even found in sewage water. So we have at least the confidence that these two MAG7 target, its original identification, because here we just show the similarity that they have, at least in this big enzyme family tree.
Okay, but that's my question was in that direction, because okay, it's not easy just to arrive there every time that you need, or just to identify. So if you have some kind of family or related enzymes that are, or sorry, bacteria or species that are more easy to cultivate and to use, so then the application could be more, more useful.
Yes, exactly. But once we publish the amino acid sequence of this, it could be easily used for E. coli expression, for upscaling, for bioreactors. Because the plasmid we use here, it worked perfectly well with the expression system in E. coli. Yeah.
Okay. Okay. So thank you.
Thank you very much.
We have just time for a very short question.
Yeah.
So I just wanted to ask you, in their natural environment, what kind of polymer the enzyme, the bacteria would use those enzymes for? Yeah.
So since our site is a hydrothermal vent, naturally it's very rich with hydrocarbons, and most of the plastics are mainly derived from a fossil fuel. And these hydrocarbons, of course we are not sure if these the species of this really utilize the hydrocarbon from a hydrothermal vent, but we don't know if it is expressed once it's available, which still, because it needs further investigation. But these enzymes, microorganisms tend to have this kind of enzymes in their genome that could be used if there's a scarcity of other nutrients. For example, FAT6 is a Vibrio microorganisms. In the Vibrio genome, there's a study conducted by one of our collaborators where they locate the location of these plastic degrading enzymes in different species, and it's there. But right now, we still don't know when we'll reuse it, something like that. But it's there. Yeah.
Well, thanks a lot.
Thank you very much.
To the audience, and it's time for a coffee break. Yeah, the sound is
This one is not mine, it's Fran's.
Okay.
Mine is the last one.
the PowerPoint one. Okay.
But you need to rename.
Oh, okay.
It was not, it's not, you need to unzip it, so you need to go up and unzip it, I think. Yeah. Yeah, that's one.
Ah, this again, come on,
Escape.
escape.
Escape. And then,
I'm going directly on the laptop for
No, you can swipe on the,
Yeah, you can.
the best thing is so that people online and the jury sees the screen, is you stand to on the side, and you can just swipe like that, don't push, because that will write, just swipe.
I'm gonna try.
You can try it now,
No, wait, I
back
think
and
the,
forth.
the swipe is not connected.
Ah.
Making a lot of
I'm
noise.
not sure if he will like it, but
Can you try?
can you try now?
Yeah. Now you don't need to go to the other side to swipe back, you just.
Oh, I can do like both.
Yeah, you can swipe both ways on this stand. Alright, so welcome back everyone. We are gonna follow this morning of presentation with 3 more presentations in this room. Alizé Jamon will be the first student to present her work. A reminder, you have 15 minutes to present. When you come to the 12-minute mark, I will wave 3 fingers in the air, meaning you have 3 minutes left. If you reach the 15 minutes, I will stand up, and that's a clear sign for you to wrap it up quickly. So, the floor is yours. Thank you very much.
Good morning. So I'm Alizé Jamon, and I'm gonna present my master thesis that I did in the CCMR in Portugal on the biotechnological valorization of the invasive microalgae Regulopteryx ocamerae in a biorefinery context. So, not like that, that works a bit too fast. So I want to come back first on the problem of marine invasive species. So there are like non-native species that are disturbing the marine ecosystem, and usually they have like 2 pathways of introduction through human activities with shipping and aquaculture. Nowadays we have over 80% of marine ecosystem worldwide that are affected by these marine invasive species, and they can create different impacts. So either ecological by disturbing the native biodiversity with direct or indirect interaction. Economic impact also because they damage fisheries, tourism, and even sometimes infrastructure. And environmental because they can alter or reduce habitat and or water quality. One of these invasive species is Regulopteryx ocamerae. It's a brown algae, quite small, so 10 to 20 centimeters, but it's forming really dense mats, and it's coming from the Northwest Pacific Ocean, specifically from the Japanese coast, and it can attach to any rocky substrate. Normally, we find it in shallow subtidal zone, like 0.5 to 5 meter deep, sometimes to 15 meter deep, and he's really rich in polysaccharides, polyphenols, and secondary metabolites. As I said here, it's really invasive. Here you can see a photo taken in the Mediterranean Sea. Everything that is brownish green, it's that algae. The introduction of Regulopteryx ocamerae, we think it was through ballast water coming from Asia, but it was totally silent until 2015, when we started to really see an impact of this algae, and it was like coinciding with a peak in surface temperature, so we may think it's linked to global warming. After, like, this species is also smothering other algae and completely destroying the ecosystem. The expansion was quite fast along the coast, as we can see, like 2015 until 2019. But currently, it's even worse, as we can find it like in the north basin of the Mediterranean, along the coast of France, in Italy, and even in Portugal, where I worked. So that species is really covering the beaches with old algae, and it's clogging the nets. So as I said previously, it's causing a problem for environmental, but also economic structure. I forget something, now we also find it like to 10 meters to 20 meters in the subtle littorals, in going deeper, so it's affecting even more ecosystems. How to deal with it? So through the bioraffining concept, it's an holistic approach to try to use a biomass into the cycle, like for industry, and after the waste is like integrated again or used for something else like recycle. Here the waste is also the algae itself, because the algae are considered waste, but it's a biomass. And the bioraffining concept will be implied in extracting the molecule in that algae, and after each extraction, trying to do another one until the final step that could be enhanced composting or something else. So the problem of managing this species that is disturbing the ecosystem is controlling its speed, but also the management strategies nowadays are not efficient, and they can even cause problems, like with the traditional removal method. And for that, we can use the bioraffining concept, like a valorization using the biomass, like identifying like the valuable activities by sustainable and eco-friendly methodologies. The point is to reduce the environmental impact of that species and also create economic value and contribute to a circular economy. For that, so I worked on multiple successive extraction using like the bioraffining concept. So it was like extraction using different solvent with different polarity. So water after methanol acetone, ethyl acetate, glychromethane, and hexane. Every time we keep the extract, and the remaining biomass is implemented again in a new extraction. And also the residue is also kept at the end. So in biotechnological domain, we have a lot of assays possible. These are the main ones, but due to fundings and also time, I'm only focused on antioxidant, metal chelation, enzyme inhibition, the mineral and CHN analysis of my residue, and biostimulation. So I want to come back on the importance of antioxidants, metal chelating, and reducing, and enzyme inhibition for industrial purpose. So here are the possibilities with the scavenging of free radicals, so antioxidant by seaweed polyphenols, but also the iron chelation by fiber or flavonoids. So these are like to reduce the rust, so reactive oxidative species that are damaging so the cells or like creating like carcinogenesis or mutagenesis. Another part is the enzyme inhibition activity. So by inhibiting so the cholinesterase, so acetylcholinesterase and butyl cholinesterase, we can participate to finding like new neurodegenerative treatments such as Alzheimer's. And the last one is for like the tyrosinase inhibition. So tyrosinase is part of the mechanism of producing melanin. So by inhibiting it, we can use it in cosmetics. So after, for our residue, we want to know if we can still use it. So it's the importance of minerals to support like the health system with dietary supplements or even animal feed. but also to know if we can use it like for soil fertilizer or as like a plant biostimulant. Also, there is like the importance of the CN ratio for composting at the end of my bioraffining process. An optimal CN ratio really ensures like an efficient composting process, to prevent like the nitrogen loss and promote like the microbial activity. So last is for plant biostimulants. So in Portugal, where I worked, we have a lot of Gulf, and we wanted to see if we can use that seaweed as a Gulf third grass biostimulant. Actually, I still don't have the result because I had a fungi invasion in my experiment, so I'm still working on this. So for each of my assays, so for the antioxidant metal chelating enzyme inhibition potential, I use spectrophotometry as there are protocols using colorimetry changes. Microplasma atomic emission spectrophotometer was like to determine like the mineral content of my residue, and the CHN elemental analyzer measures the composition in CHN, and that can give us like the protein content and the CN ratio. Here are my results concerning the radical scavenging, so antioxidants, metal chelating, and enzyme inhibition activities. So we use the EC50 or IC50 is the half maximum effective concentration or half maximum inhibition concentration. That means the concentration we need to attain half of the inhibition or to stimulate the antioxidant potential. It's made like to be able to compare the result with the standard and also all the other values from other. So I put in red the values that are smaller than the standard used in the industry to really see that like the water extract, the ethyl acetate extract, and hexane extract show really a good potential for antioxidants and enzyme inhibition. When I say smaller, it's way smaller. It was significantly smaller. Sometimes we also don't have any possibility to compare these results because there is just no study on those actually right now. I want to show also that for the ethyl acetates, we have like a lot of antioxidants, metal chelating, and enzyme inhibition. And that could be linked directly to the total flavonoid content. Here, as we can see, we have nearly 256 milligrams per gram of dry extract. So it's really high, even for a lot of dictyl, that is the same order of brown algae, we don't find as much. Concerning like the flavonoid, phenolics, we find like the same value. So we can think for ethyl acetate, because the polarity is more neutral, it can extract more compounds like flavonoids and phenolics that usually are linked directly to antioxidants, metal chelating, and even inhibition acids. Here are my minerals in the residuals. So first of all, I want to attract attention to those, so the cadmium, the lead, and the chromium. So they're under the critical levels, so we can use it. It's really important because if it was over, like even the use of this algae, it's not possible at all, and the residue, it's not possible ever. After, we still have a bit of potassium, magnesium, and sodium, so it can maybe be used in soil amendment, even though it's really low, but it can be implemented into another product with other species, or like even a fresh algae. Concerning the CHN analysis for the residual, so we saw that like the optimal range for composting is not attained, but by mixing it with carbon-rich products such as like straw or wood chip, we can attain that optimal decomposition rate with enhancing the carbon. For the crude protein content, we have 16.5% of crude protein. So it suggests that we can implement it into animal feed. as like a supplementary protein source. Because nowadays, using only microalgae or protein source for animal diets is still not completely done. I want also to show the decomposition considerations that we need to have. There is a lot of high sesquiterpene content that is toxic, so we can't use directly vermicomposting. But there were like methods that show that black soldier fly larvae and vermicomposting, so with cockroaches, was possible. So we can do a full bioraffinery process here. So for the conclusion, as I said before, the ethyl acetate extracts show really important activities for the antioxidant and metal chelating and also enzyme inhibition. There was also a possibility for the water extract for the nitric oxide scavenging. The hexane was interesting for the metal chelating activity. And really the ethyl acetate again with ACHE, BCHE, and tyrosinase, so it can be used like for neurodegenerative disease treatment and cosmetic. After the chemical composition of my residue really shows that we can do soil amendment and composting after this. For the next step, we really need to optimize the extraction method to enhance the bioactivity. Here I worked on an algae that was like took from beaches, and after it was dried, first rinsed with water, dried and froze. So maybe working on that part of the methodology would change a bit the activity that we have, but even on an algae that was not fresh, we still have a lot of possibilities and activities. After, we also need to determine which molecules or complex is responsible for which activity, because we want to explore the commercial application for it in pharmaceuticals, cosmetic, and agriculture, to implement it in the bioraffinery context again, to find a value for that species that is invasive, and we want to try to use it industrially to remove it from the ecosystem and manage it like that. And whoop. Again, sorry. And here are my references. And do you have any questions?
Thank you very much. They're pretty much on time, about like 10 seconds before the end of the 15 minutes, so that's pretty good. So, any member of the jury wants to go first?
Okay, I'll go on the end of the table. Yes, with pleasure. Thank you very much for your presentation, and I think it's very relevant to be looking for such type of compound and use of algae, considering the global issue related to harmful to those blooms of macroalgae and this species and others. My question, you have been something, I would say, I guess, your sample were coming along from the Portugal coast. Do you have any sense on how sample or algae from other parts of the world, would they have the same value? What's your thoughts on that?
Actually, I compared a bit with other type of studies that were done on that species and also dictol. And the one like invasive right now in the north of the Mediterranean basin and in Portugal are quite the same. So like even like genetically, so you're not different for now. So like the activities is the same.
Okay. And in terms of contaminant, I cannot recall which one you have analyzed.
For the contamination, you're talking about like the mineral contents.
So you had cadmium, yes. So even cadmium was below the.
Yes, they were all below the critical levels. So, but still, because like I was talking after about the sesquiterpene, that is a toxic terpene, we still need to do toxic assay for this algae to be sure, like after, because before doing a drug, we have a lot of time because we need to be sure it's not toxic, and after doing the drug, and that the drug in itself is not toxic. So here is like I will say is the first part of using that algae. After we need to do like a lot more research on it.
So what can you tell me about this? Now it's a little bit off your work, but what can you say about these critical levels? In terms of, usually there are sets based on consumption.
Yeah.
So if you propose to increase the use of such compound, what consequences it can have on these critical levels, and do you think it can in the end impact somehow human population or animal?
If we using that algae in diet.
Yes, for example, as diet or as feed for.
So first, when we implement it in health supplementary or like animal feed, they're always going like through filtration first, and after like there were like more tests. So it's not like directly using the raw product. Here I'm only working on the raw product. So if we are under the critical level with a raw product, being under the critical level for the final product is like quite sure, I would say.
No, we have time for another question. Yes, for the preparation of our extract. So I've seen you're doing successive solvent extractions, and then how did you proceed? So you have your solvent. You collect the solvent. I mean,
So I have my solvents, so I choose them depending on polarity and also like the molecules that were found in other dictol. So those were the solvent used normally. So every time I'm doing like my powder, I'm putting it like for a cold maceration with movements, and after I'm doing a filtration, I'm keeping the solvent there like with the extracted molecules, and I'm drying the remaining biomass. I made sure that like each biomass was like completely dry. It was like easier for like with hexane because it's really volatile, but like before it took a lot of time sometimes. And after the extract went through a rotary evaporation to make it more concentrated. After this extract was stored in a fridge, like it's a lab fridge at -20 to keep the extract. And after, when I wanted to do my assay, I resuspended them either in the solvent or DMSO, because for some assay it's not possible to work at all with the solvents. As I say, some are like completely inhibiting the assay, or they just like really volatile. So when you put them in like the plate, they're all gone in like within 3 minutes, not even 3 seconds sometimes.
so my question is, how can you control in your assay, because after you measure the activity, what matrix effect you might have, and how confident are you in your analysis in terms of positive or negative, or quantitation of any compound you might have?
Right now, until the article is out, I can share any raw data or things like that. Also for me, it was easier like to present everything as you saw with the colors, because if I show only the numbers by themselves, they're not really talking or saying anything by themselves. And if I understood the question.
I just want to know if you have any is there any possibility that you would have any matrix effect, meaning that your measurement might not be the actual measurement of an enzyme activity, or how do you assess that?
I did technical replicates for each assay, so to be sure it wasn't right. Even like for the total flavonoids that were like really high, we were like, what is going on? Is it like an error? Sometimes like it can happen, but actually I did 3 times with technical replicate inside those 3 times. So we know that for the biomass I had, these are like really corresponding to the solvent I used. After, for the comparison, sometimes people are not using dichloromethane, so I can't compare to the activity I have with my dichloromethane extract.
I would like to ask about the polyphenolics, the antioxidant. Are you aware of the literature where you look at the polyphenolics in a more ecological way?
Yeah.
Have you
For some parts, yeah.
Sorry?
For some parts, yeah, because like for some they're like studied a bit like for the dictol order, and for some others they are not studied at all.
Yeah.
Even for the flavonoids, some molecules like there aren't any study in the dictol order, so that's why I couldn't like compare them at all.
But in the few products, for example, we've used them for a long time in a more ecological way, and you had like 4% 40 milligram per gram.
It was in my residue.
But if you look at the literature that we have from an ecological point of view, it can vary from a couple of percent up to 20%.
Yeah, it's like sometimes like
Yeah.
Dipol, I have it at the end. I'm gonna go there.
Yeah.
It's going to be faster like that. Here. So actually, like for the phenolics, in the Dipol order, it's like 2 to 25 usually.
Yeah. So, but it's also from an ecological point of view, this system can be inducible. So animals can induce this system because they use it as a defense.
The thing is, here we are an invasive algae.
Yeah.
So they don't have any predatory
but I
algae
was
yet.
thinking about when you would like to use it to optimize the content, you can actually look also to the biology around it. Because if you want to have a lot of polyphenolics, then you should maybe take it in a certain time.
There is that part, but also I want to really show that we're on an invasive algae. So the point here is to use it directly from beaches to remove it. So not to try to industrialize the way we're taking it, because after maybe people will want to really rely on it, and they will want more of this algae again and again. So it can be like the dark side of it.
Okay, I understand. Thank you.
Baron?
Yeah, no, some questions. Okay, congratulations for the presentation. You explained well that. That's very understandable. And yeah, one of the questions were related with that. No? Okay, so you are doing your aside in invasive species. You almost answered what I was thinking of. Okay, try to give a biotechnological use, but at the end, it's an invasive species, so that can produce that people grow that and so on. Okay, so the solution is at least difficult, you know.
Actually, I can talk like for Spain and Portugal right now. So we had like a link with the University of Malaga and even a startup in Portugal. In Malaga right now there is no permit to gather that species as an industrial product. It's only possible to take it to do research on it. And in Portugal they have a permit to gather it, but they need only to take the one that already is on beaches or clogging in the nets. Here, like same like for Spain and Portugal, this algae is considered a waste. So they need to remove it from the beaches, but the only thing they're doing is burning it. So here is trying like to find a way of not just spending money on it.
For example, there is any other species, local species, similar of the same genus that can be used? Or can it be extrapolation of your results?
I didn't look at the local species. So for that, I'm not sure. But if the point is like to try to use an invasive algae as a value directly, using a local species that is already like bad, in a bad state, like for the marine environment, is not a good idea, is it?
Not a idea. It could be like a strategy to find to push this local species because it's of the same genus or family of this invasive one. So we can assume that maybe.
For the dictol, like most of them are really in the Japanese coast or the US coast. Right?
And I think that molecularly you only characterize the flavonoids and so on.
Actually, I didn't
No?
characterize it. It's just like a total like flavonoids content or phenolics content. That's why I was saying like the next step is to know which molecule is it really doing what activity, which activity.
Oh, okay.
Yeah.
So
It's
you
like
plan to introduce all the biomolecules, not only.
we need to know like even like for I was saying like the water extract for the nitric oxide, we need to know which molecule is really creating that antioxidant potential. Because like for the industry, they never use the raw material directly. They're extracting like one molecule and filtering it. But after we need to use like HPLC for that, for example.
Okay. because my impression is that sometimes as you are using raw extract, let's say, so
Yeah.
it's a combination. Maybe there is not like one molecule that you isolate and it's,
Yeah, that's it.
you can say that, oh, this reduces acetylcholinesterase, this,
Yeah.
no, it's probably you extract there are more than one that
Yeah, it's a
interact.
complex.
Yeah.
Good. All right, thank you very much.
Thank you.
So we need ask Francesco to get in. Francesco, if you can try to like stand on this side of the screen. But because otherwise the people online will not be seeing you. Because like if you have friends and family and also for the recording, if you don't want to be seen, you can stand on the other side. But yeah, that's better I think if you do. So you have 15 minutes.
So I'm going to present my master thesis about the analysis and utilization of vulva rigida, palmaria palmata, and fucus vesiculosus for potential dog and cat snacks. Okay. So thankfully for the studies carried out annually by the European Pet Food Federation, we can see the number of pets in European households. So in 2021, there were 310 million of pets in the European households, and in 2023, 352. That's an increase of 13.5%. And in specific, in Portugal, we can say the data that the increase is 28% from 4.59 millions of pets to in 2023 to 5.87 millions. Also, thankfully for this federation, we can see that the market of the food for pets, it's more in 2023 was more than 29 billion euros per year. Among all the ingredients that they use for the diets of the pets, we choose algae to create algae snacks for them. So the first part was the algae. So we try to find algae, one green algae, one red algae, and one brown algae. Algae that are consumed for humans, already consumed for humans. So in green algae we choose vulva rigida because of the antioxidant power and the nutritional composition in proteins, lipids, among others. In the red algae we choose palmaria and palmata because of the nutritional composition between proteins, lipids, minerals. And in the brown algae because of we choose fucus vesiculosus because of the mineral content and the anti-inflammatory power. So for the snack part, we worked with the company named Babe Michael Happy that they do snacks for cats and dogs in Portugal. So we were looking at snacks because the snacks has a complementary role in the diets of the dogs and cats. They are good to complement the diets and they are healthy for them. And we were looking for some key parameters like antioxidant power, enzymatic inhibitory properties, anti-inflammatory properties, and the content of protein, sugar, lipids, and mineral content. Also, how I mentioned, the snacks health benefits, like they boost the immune system, they improve digestion and ensure balanced nutrition with essential nutrients. So the objectives of this study was to comprehensive analysis of the bioproducts of these algae, formulate and produce pet biscuits or cookies incorporating these algae. Also, we make an evaluation of acceptance in dogs and cats, and we create a survey for the consumers to see if they like the idea to give to their dogs and cats algae snacks. So I made a graphical asset just to make an easy follow presentation. The first point is the nutritional profile. For the nutritional profile, start with studying the total proteins content. There are two graphs. The biomass graph is the graph of only the algae. The algae were dried, and the cookies graph.
Yeah, because you tapped it, so it advanced twice.
Okay, so these are there are significant difference between ulva and fucus and between palmari and fucus, and there are no significant difference in the cookies ones. Also, I compared the results in all of my essays between the cookies and the biomass to see if there are differences, because the cookies are made of algae that we are going to see later. So also we check the content of sugars, and we can see the significant difference in sugars in biomass with ulva and fucus, like previously. And in cookies, there are also 3 different groups that they have significant difference. And also the comparison between algae and the cookies results. Then we make a moisture, we put the biomass, the powder of the biomass and the cookies in an oven, 105 degrees for 24 hours. And the results are these ones, with the significance and the comparison between them. There were significant difference when I made the crustal, but then when I checked the post-hoc done with Bonferroni, they give me that there are no significance. It can be because of Bonferroni or because in crustal they are doing also comparison between cookies or between algae, but we don't want that, only one algae and cookies comparison. Yes, was also we put the powder in an oven for at 505 degrees for 4 hours. In the comparison, there were significant differences between all of them. And lipids, I did the lipids to see the lipids content. There were no significant difference between in biomass, and there were only significant difference between the control cookie with the palmari palmata cookie. And the comparison between them, it was the same as moisture. There were significant difference in crustal, but no significant difference after the post-hoc. Then for the minerals content, I have 2 groups, that's the macro minerals and the trace elements. For macro minerals, I also put the cookie control in other group, just to see the results. The mineral with more amount in biomass was potassium, followed by magnesium, as in the cookies, but in cookie control was phosphorus, followed by potassium. The trace elements, the mineral with more amount was zinc, followed by manganese. In the cookie control was the same, it only the only differentiation is the lead here. And in cookie control, in cookie was manganese with zinc. Okay. And the last part of the nutritional profile, also check the pigments of the biomass, the chlorophyll A and chlorophyll B. There are significant differences in Ulva richida and Palmaria palmata in both cases. And then in the total carotenoids, there are significant differences between Palmaria palmata and Fucus vesiculosus, but probably it's because there are an error here in this study. The second point is the extraction. I made the extraction for the following assays. We take the powder of the algae, we mix it with methanol, then we filter it, and with this filtration we'll put it to evaporate overnight. Then with methanol we make a concentration of 50 milligrams per liter, but then we dilute to 10 milligrams per liter. After seeing the results, we see that we want to use the 10 milligrams per liter. The third point is to check the antioxidant activity. So we start with the DPPH. There are significant difference in the DPPH between these three groups, and then we check the ABTS, that there are significant difference with these groups, within these groups. Also we check for the antioxidant activity the CCA to see if there are, we see that there are significant difference, and with FRAC. There's only significant difference between control and Palmaria Fuc. No Palmaria. And then the fourth point is the TPC and TFC, phenolics and flavonoids. We did phenolics and flavonoids because they are a good indicator of anti-cancerogenous, antioxidant, and anti-inhibitory. And we can see that there are significant differences in phenolics between these three groups, and in flavonoids between Palmaria and Fucus. The 5th point is the enzymes, to check the inhibitory enzymes. So we use ACHE and BCHE, that they are good indicators of the inhibition of enzymes. Now we have the significant difference, the other graph. And then I was talking all the time about cookies, so we are going to see how we make the cookie preparation. We use flour, eggs, biomass, algae, and water. We mix them in different proportions, and we did the shape that we want, and we put it in an oven for 20 minutes. And we have the control without biomass, and the other one, Algae-based, Palmaria palmata, Fucus vesiculosus cookie. And then the mixture is a mix of these three with the same quantity of them. This is an image of the cookies. And then we wanted to know if the dogs and cats like the cookies or not. So we went to a shelter in Faro, in Portugal, and give them some cookies to check them. In the cookies to 17 dogs, only 2 of them didn't like them, but they were a bit afraid, maybe because something in the past, so maybe they don't like because of that. And the other 15, they like the cookies. In the case of the cats, all of them like the cookies. There are differences in each cookie because I cannot give all the cookies to all of them because there are few, so I had to split it with another cookie. And then we make a market survey on the Instagram of the company. So we ask them, to the consumers, if they will you buy biscuits made from seaweed. 67% answered yes, 50% answered maybe. So it's a high response, high positive response from them. So for conclusions, the algae, in the scientific part, the algae and the cookies have some high antioxidant potential, enzyme inhibitory activity, and chelating properties. The acceptance, dogs and cats show great acceptance and interest with the cookies, and the market, the consumers like the idea to give cookies to their dogs or cats. For future perspectives, maybe it's a good idea to put other algae that like spirulina or nori in the mixture with the algae that I studied, because spirulina in this case is high in proteins, in vitamins, minerals, or nori. Also, it changed the flavor of the texture because they were a bit rigid. So maybe using fresh algae, they improve the organoleptic properties. Then change the preference. I made a preference testing. We can give 4 cookies and they can choose the one that they prefer to see which one they like more. And the last one is improving the acceptance process, because in the case of cats was not the same number of individuals, and also tried in different households because all of them came from the same place. That's all.
Thank you. Thank you very much, Francisco. 14 minutes, 30 seconds, so basically on time. Who wants to go first? Mary again? Okay.
Thank you for this presentation, trying to make food healthier for cats and dogs and other animals. I would like to know, your cookies were they made 100% of algae, but mostly what I would like to know is what was this control cookie, how did you choose it, and why do you call it a control?
The presentation is 75% flour and 25% algae.
Okay.
And the control is without the algae.
Everything without the algae. Okay, I have this information. And when you say mixture, it's a mixture of all the 3 algae?
In the same proportion.
Okay, thank you. And you mentioned in many of your tests you're interested in measuring the moisture, the ashes. Can you tell us why you do that?
Yes. For the moisture, with the moisture, because you can see if the food is going to have a long life, like
Okay.
when you open the pack and all this. And the ask normally is to see the inorganic minerals.
Okay. Had been yes, great if you had provided explanation. And so you have done a lot of statistical analysis to compare everything. But I wish you had provided us some interpretation, further interpretation. So quickly, so which one would be the best algae?
I'm still doing that, but
you're still analyzing it.
yes, but in some cases it's focus.
It depends on the.
Yes, but also the Palmaria gives some. I don't remember which one, but gives some properties that the other ones no.
Okay. Thank you.
I could continue. If you would like to scale this up and sell them, and think, what do you think about species? Can you get the biomass from all of them easily, or you would like to scale it up and sell this to millions of the dog owners?
Okay, also I asked that to the company because we were working with the company because they are already doing it, but not with algae. So I said that, and they told me that they don't have in mind to do something big. It's only like more national in Portugal, and the market there is big, but it's not that big to be a problem.
So it's easy to even get ulva, the biomass from ulva. I guess fucus is easy to get, and Palmaria you can get because it's commercial, but I think ulva is like thin sheets, and you need a lot of them to get the same.
The thing is, these three are from also a company from Portugal,
Yeah.
and they already
So they are commercially sold,
Yes.
so you already get it.
yes,
Okay.
from a company from Portugal.
Okay, thank you for the presentation. I have one question about the enzymes that you determine, because maybe I missed. Okay, at some point. No. Okay, you determine acetylcholinesterase, and so my question in this case is acetylcholinesterase. What is the information that you want to obtain from that when you say, okay, that an inhibition is a good signal? Because this is controlling neurotransmission.
There are accurate inhibition of.
Yeah, you say that in your conclusions, as far as I understood, that this is positive. It's a positive point that the algae extracts inhibits the activity of this enzyme.
So I understood it's if there are good inhibition of the enzymes, it can be good for the immune system, for the health of the animal.
Yeah, but I mean that this enzyme is essential also. So, for example, your control. What is your control? I mean, like what? Okay. So I mean that at some point this needs some other view also. Okay, in order to understand you are inhibiting this enzyme, but at some point it's also essential. It is true that there, that some contaminants can stimulate, but some contaminants also can repress this enzyme. Okay, and you need to balance it with other complementary analysis to see that what you are observing is positive at all. At that point, to be treated.
I also did another enzyme that was alpha glucose, I think. But we had a problem with it, and we were waiting that a company sent more, and we didn't receive. So it's because of that I didn't put the small information that I have.
Okay. Related, for example, just with the test that you did at the end, you say that they eat happily or not happy. Okay, I mean that. How you test it? I mean that you give the cookie and so.
Was just to check the acceptance, if they eat the cookie or not. Was not to check like in how I talk in the future steps to check if they like more the vulva or the fucus. We didn't check that. We only check the acceptance if they eat the cookie or not. Only that.
Okay, and you also mentioned that maybe instead of using extract, using the fresh.
Yes.
Okay, in order to prepare. It needs analysis, but I don't know if they can accept something, if they are not used to it, something coming from the sea.
No, but it smells like fish, so they like fish, and cats for sure, but in dogs also. Because it was like a question that we had before we did the snack acceptance. We were like, it smells a bit like the sea or like a fish, but they like, we talk with the people that they are there with the dogs and cats. They told us that there are no problem with the veterinary also.
Okay, I was wondering about because one thing is fish meat and then algae. Okay, and it's the textures and so on and so different. Quite promising. Okay, thank you.
Thank you. Just a quick question.
Yes.
In many of your graphs, there's a difference in the relative values between the species, the originating species, between the cookies and the raw. What in the process explains that? Do you have any idea why it changes the relative values from the raw algae to the cookies? Because I don't think, unless I missed it, I don't think you ever mentioned that. Why does it change? For any, like.
Why we didn't check the other essays in cookies?
No, I mean, like here, the value is the largest for Parmata, where when you go to the cookies, it's the lowest. And why does it change between the raw algae and the cookies? Do you have any idea? Do you have any explanation for this?
No. Maybe it's because of the other ingredients, but.
Okay, all right. Thank you very much for this. And now to set up, we're gonna wait just 2 minutes and also that we start on time for the people online. So we're gonna start at noon sharp.
Is this your I don't know why it doesn't open.
Can I send the green?
No. Okay, you know what? I will ask.
What? Did you get the message that Elise got before? Laurence?
Sorry?
Are you getting the same error message that Elise got?
No.
It's not working? Or is it?
No, this one is not working.
Copy it to S1, because that seems to solve the problem for
Okay,
Elise.
so.
Earlier.
Okay. Where is the S2?
Oh, it's a RAR, I'm not sure. And it doesn't show an icon. Right click on it. No, it's a RAR. It should be a zip, I think. Because this computer is not equipped to unzip that.
Okay. I will ask Tim.
Yeah.
I copied it also in the S1.
No, there's no.
There's no.
It's not zipped.
Short intermezzo.
Alright, very good to know you have 15 minutes.
Okay. Hello everyone, thank you. My name is Arnold, and today I'll be presenting my thesis with the title Nutritional and Bioactive Potential of Atrospira platensis, Chlorella vulgaris, and Phytoremediation limacinum for pet cookies application. The thesis was supervised by Dr. Luisa Custodio from the University of Algarve, Center for Marine Science in Portugal.
You sweat.
So the study is actually stemmed by increased ownership of pet globally, where in EU we have seen increase.
Sweat, not fat.
Yeah, we can see increased dog ownership by 57.58% from 2017 to 2022, and cat ownership increases by 71.62% from 2017 to 2022. This staggering number is high, but current feeds market relies on first animal byproducts. These are usually the animal leftovers that are eaten by humans and then are given to the dogs. And often they lack those nutritional components that the pets needs. Also relies on grains and cereals. Grains and cereals are great, but they are usually used to provide energy to the cats and dogs. However, to most of the dogs, they usually have associated with issues like allergies, and they're not fully nutritional for pets and cats and dogs. Also, we relies on synthetic additives, and these synthetic additives usually added to those feeds to preserve for longer terms, and also they're used to add flavors. But research have shown that synthetic additives are usually associated with effects, long-term effects. So, by these problems that we have, we need sustainable and nutritional balance alternatives that will be provided to the market and fill these gaps. And in our studies, we are going to rely on microalgae, and these three species that we're going to rely Atrospira platensis, which is also known as Phytoremediation, and Chlorella, and we're also going to rely on Phytoremediation limacinum. So why are we going to use this species? First, for Atrospira platensis, is high in protein. Research shows that it's 50 to 7% protein, and also composed of essential amino acids. It has high composition of phycocyanin, and phycocyanin is antioxidant compounds, and the antioxidant compounds can remove those antioxidants that are found in can remove those oxidative reactive agents that are found in the cells. It's composed of vitamins, minerals, and essential fatty acids, which are also very important for the cats and dogs healthy. We are also going to, we chose Chlorella vulgaris as Chlorella because it's rich in chlorophylls. Chlorophylls can also be antioxidant compounds, and they can use to maintain the healthy status of the animals. It also composed of proteins. So research shows that it's 50 to 7% of proteins. And for Phytoremediation limacinum, it's rich in DHA. This other research have shown that it has been used as alternative to fish oil because it's composed of omega-3 fatty acids. So the objective of this study, first, is to determine the bioactivity of this species, and then after determining the bioactivity, we're going to determine the proximate composition, including the protein, lipids, fats, ash, and moisture.
Our second objective is to develop this microalgae, either first individually, one by one, and also we're going to mix them. We mix them to have the combination of all three microalgae in the same cookie. Number three is to determine the proximal composition of these developed microalgae. Objective number four is to determine the palatability prepared to the dogs and cats. And then objective number five is to know the public interest, how the public perceive this microalgae for their pets and dogs. So methodology, the first one, we took the microalgae biomass, and then we extracted using methanol, and then after extraction with methanol, we analyzed this bacterial composition, including the copper collecting activity. We determined the FRAP activity, which is ferric reducing antioxidant power. We determined the ADTS and DPPH. This represents the radical scavenging activities, and also determined the enzyme inhibition activities, including the acetylcholinesterase and butyrocholinesterase. And apart from the bioactivity, we determined the composition, which is flavonoids and phenols. And also we determined the pigments, including the chlorophyll A, chlorophyll B, and carotenoids. And after the analysis of this composition with activity, we determined the approximate analysis, and that we're going to see in the next slides. So for the cookies, we made two ratio. Using two ratio, the first one is 50-50. That means we use 50% of scald flour and 50% of individual microalgae. And for the second ratio, we used 75-25 ratio. But according to analysis that we done in the lab, we found that 75-25 ratio has good texture and composition. So we continue with 75-25 ratio. For proximate analysis, we, as I said earlier, we determine the protein concentration, lipid, total soluble sugar, and mineral content. And for the protein, we use, and for all the methodology, we use this methodology as shown here. Also, we determine the moisture content and ash contents. And for palatability tests, we asked 3 different questions. We did a study to know 3 different parameters. The first one is initial interest. So for the initial interest, we'll see later how we structure the question and how we got the results. And also we determine the eating speed and subsequent interest. For public interest, as I said earlier on the objectives, we asked the question, we did the survey, and the question was like, would you buy cookies made from algae? And this is the cookies that we made, as seen on the photo. So let's dive in on results. And here are the results by activity of microalgae biomass, as seen here. So the first column is represent the metal collecting activities, and represented by FRAP activity and copper collecting activity. And we can see here that atheroscleral chlorella vulgaris was more potent, and this is because it has EC50 of 0.72. The lower the EC50, the higher the potence. That means it has more antioxidant power than other species. So for the radical scavenging activity, we see here that ADTS was more sensitive in recognizing those radicals compared to DPPH. Also for enzyme inhibition, we can see here that S-limacin it had IC50 of 1.76, which is more effective compared to other microalgae. So for the flavonoid and phenol and chlorophyll, these are compositional parameters that we use in our experiments. So we can see here that Athrospira patensis was significantly higher compared to other microalgae. And the high composition was seen in flavonoid and phenols, even in pigments. We can see here that it was higher than Chlorella vulgaris. But however, for the carotenoid, we can see here that we have lower values of carotenoids in all three species. We'll discuss later why, but this might be attributed by our methodology that we used. It might be caused by the dissociation of these carotenoids while doing the experiment. So for proximate composition, I want you first to look on this mixed cookies. So here we can see that the total solute protein on mixed cookie had high concentration of protein compared to other three cookies. Looking on ashes, also we can see here that there was high concentration of Athrospira patensis. However, in lipids here, we can see that we had generally lower percentage of lipids. For the mineral composition, generally we had high concentration of potassium, calcium, and magnesium. That is for macro elements. However, in micro elements, we had high composition generally in iron compared to other micro elements. For palatability test, as we see here that, as I said earlier, that we had tested three questions. The first one was initial interest, as seen here. So there was no significant difference when the pets were given these cookies. Looking on eating speed, also all the cookies were eaten the same speed. However, in subsequent interest, we see here that there was lower interest in cookie made with S-limacinum. For the public interest, generally a high majority of people showed high interest for the cookies. Other 30% says that maybe they would consider to buy it, but according to the papers that I read, it says general knowledge of people to this microalgae benefits can result to increased number of interest. So for the conclusion, I'll say that microalgae contain bioactive compounds, potential for the health improvement of dogs and cats. Also, microalgae supplemented cookies are highly palatable, as we have seen earlier in the last slide. Consumer interest, there is generally presence of knowledge for benefits of microalgae to the public, and that's why we have seen a high number of people who are interested to buy those algae. So future studies, I would recommend to do the long-term effects of microalgae supplemented cookies, because we didn't do that. We only looked on the composition and the acceptance, but we didn't go on to see how these microalgae influence the healthy status of pets and dogs and cats. So these are my reference. And I would like to thank this institution. It's Centro de Recolha Oficial de Faro.
And then I would also like to thank Tiago Braga, he's from Make My Dog Happy, and my supervisor, and also coordination team of MDRC. Thank you.
15 minutes sharp. Perfect. Thank you very much. I'll have the members of the jury start with a question. Mary?
Yes, thank you for this presentation. So we do see the interest as we have the presentation on algae in pet food. In several, on one table we were comparing with other algae, but I did not get which algae you compared to. What was the comparison?
Okay, because I had two parts. I had the biomass, which I did only the bioactive composition and the approximate composition, only for microalgae before making the cookies. So I compared the three of them, and then I did. I made the cookies themselves, and then I compared the approximate composition without comparing to bioactive composition. Yeah.
Okay. And if we go into slide 9.
Oh, sorry. Yes.
How could you explain that the protein content, 3.7, would be higher when you have a mixture of algae, while if you have it independently, the protein content is much lower?
Yeah, that's a good question. I don't know why it's like that, but I would say there is synergetic association or relation, because maybe my theory is that when you mix them, there is improvement, like those amino acids, they improve themselves, like synergetic effects, and that's why we have this number.
Do you measure your protein through an activity or amino acid measurement?
No, I didn't do amino acid measurement. I did only the protein composition by using.
So it's a protein composition.
Yes.
So puzzled with the synergetic effect when measuring it.
Yeah. No, that's just my theory.
Okay.
Yes.
Okay. Yes. Well, I have to. Yeah, I will see. Do you have an idea of how stable? You measure a lot of activity.
Mm-hmm.
Any idea of the stability of this enzymatic activity on the different assay you have? Have you tested the stability over time of your cookies and any?
That's a good question as well, because while we do the enzyme, these enzyme activities, we had a lot of repetition, because I think it was not stable, because we did quite a few times and the results were kind of different. But always, L-cimarcinum had high concentration. Had high potency of inhibition. Yeah, that's for sure. But for the stability, I think that's a good question.
Okay.
Yeah. As you said, you didn't study the long-term effects, but is there any other literature that where they have given algae products to cats and dogs that you can at least think of the effects?
Yeah, nowadays that I've seen a little bit of new research, but most of them they are reporting the acceptance, like how are they accepting? It's not for long term. It's only for short term. And also I saw another research that was only for public interest to use those pets for their animals, but the long-term use of this microalgae to improve the health of dogs and cats, there is none.
No.
Yeah.
Because spirulina has been used as a human food source, well, not maybe food, but at least something you can eat a little bit of.
Yeah.
But I've seen some of the studies that they actually could be toxic sometimes.
Yeah, that's true.
Yeah. So how do you know that your spirulina is not toxic?
I don't know because we actually bought the spirulina and chlorella and others from the companies, and we don't know how they culture them and until they sell them. We don't know the procedure that they used, but that is true because spirulina, chlorella, and others, those are they use the nutrient that they have to produce energy, to survive for themselves. So if they are not well cultivated, that means, if there is a metal around or there is a harmful metal anywhere around, they will absorb it. And when it's eaten by people or by any other organism, that means they are taking up those metals, and that's not.
I don't think it was metals when it comes to spirulina. It was more that it became toxic, actually.
It can be.
You have harmful microalgae blooms sometimes, and sometimes even these pieces or the spirulina can be toxic.
I've also I
don't want to give. I don't have dog any longer, but I don't try to give them spirulina.
Yeah, it's true because for spirulina, it has been used widely. It's well known. It's the best. But for other microalgae, for sure.
Thank you.
Okay, thank you for the presentation and contributions for the work. One more question is about the control of the reference. Okay, why not including, let's say, an already commercial cookie or material using other kind of vegetables or more common that are used in the market as a supplement, in order to compare that what you are observing is adding a value, or it's another value to what is now in the market.
Yeah, it's a good suggestion.
I don't know if you have some information of the composition, protein level, or what is in market. I don't know with supplements using.
That's a good. It's a good point, but our initial theory to do this was only to see the composition and how the nutritional composition and bioactivity composition of this microalgae can be transferred to improve the health of animals. So it was not to compare between feeds, I would say, yes. But it's a good point. It's a good point.
Yeah, because at the end, the ones that probably nowadays they are using are more like coming from the small commercial, spinach or something like that. So thinking in adding this microalgae, it should be economically profitable, and also in a way that can give benefits, but also at this point. And I agree with my colleagues' comments. Okay, so there are other elements that at some point can counterbalance or can interact, and it should be also at some point. Thank you.
Thank you.
Right, thank you very much. This brings us to the end of this second morning session, so we'll take a recess for lunch. We'll say goodbye to the people watching online, and we'll be back at 2 p.m. if I'm not mistaken. Thank you, everyone.